On the last part of our lecture, we will talk about bacterial growth requirements. But before we start talking about bacterial growth requirements, let's talk about what is it actually bacterial growth. Bacterial or microbial growth is increase in the number of the cells. When bacteria grow on the surface of the media in our labs, they form colonies. Colony is a clone of cells large enough to be visible on the surface of the media. So how does those colony get formed? There are 4 phases in this process. The first phase is called lag phase. Lag phase. As you see, this graph is flat on the lag phase. Why? Because the number of the cells at this phase stays the same. During the lag phase, cells collect nutrients, grow, and double their mass. Cells prepare to divide. The next phase is called log phase. Log phase. You see, graph goes up. That means number of the cells during this phase is increased. Why? Because during the log phase, bacteria actively dividing and produce new cells. And then stationary phase, the next one. You see the graph is flat again, means that the number of the cells during the stationary phase stays the same. Why? Because usually during the stationary phase, bacteria don't have enough of nutrients on that media to grow and double their mass. But they have enough of nutrients to keep themselves alive. And then the last one is a death phase. You see now a graph goes all the way down. During this phase, bacteria start dying because nutrients are depleted. Every species of microorganism grows over a range of temperature, from minimum growth temperature, which is the lowest temperature that will support bacterial growth, to optimum growth temperature. This is a range of the best temperature that will support bacterial growth, to maximum growth temperature, which is the highest temperature that will support a growth. Because different microorganisms have different requirements for temperature, we divided them into different groups. For example, hyperthermophiles grow in the range between 65 to 110 degrees of Celsius. They really like high temperatures for growth. Thermophiles grow between 40 to 75 degrees of Celsius. But I would like you to pay your attention to the next group, which is called mesophiles. Mesophiles grow at the human body temperature, around 37 degrees of Celsius. And this semester, we're going to work with mesophiles a lot. pH requirements. The most pathogens thrive in a neutral pH range. For example, most bacteria grow between pH 6.5 and 7.5. For example, molds and yeasts grow between pH 5 and 6, means a slightly acidic environment. Some microorganisms actually prefer acidic pH, acidic environment, and that's why we call them acidophiles or acid lovers. If microorganisms prefer to grow in basic environment with higher number of pH, we call those microorganisms alkalophiles. Osmotic pressure. Basically, from this slide, you have to know how cells will act if you place them into hypertonic or hypotonic solution. If you take a cell and place it into hypertonic solution, the concentration of salts in solution will be higher than concentration of salts in the cytoplasm of cell. As a result, that solution will try to pull water out of cytoplasm of cell. As a result, cell shrinks. If you place the same cell into hypotonic solution, then concentration of salts in cytoplasm of that cell will be higher than concentration of salts in solution. As a result, water will be pulled from solution into cytoplasm and cell swells. Basic ingredients that bacteria need to grow are listed on the left side of this slide. You have to remember that carbon is a major chemical element in biochemistry. For example, a lot of bacteria use carbon as a source of energy. Besides carbon, bacteria require hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And the last requirement for growth will be requirement for oxygen. Because different bacteria have different requirements for oxygen, we divided them into 5 separate groups. Basically, what you see on this slide, those are tubes with nutrient broth, and inside of that nutrient broth you see bacterial cells. The first group is called obligate aerobes. Obligate aerobes absolutely require oxygen for growth. They cannot survive without oxygen. And that's why you see those bacterial cells on the top of bacterial broth. Next group, we call them facultative anaerobes. Facultative anaerobes. Facultative anaerobes is a very interesting group because they can switch their metabolism from aerobic to anaerobic. So when oxygen is available for them, they grow as aerobes. They use it for metabolism. If oxygen is not available for them, they grow and multiply as anaerobes, means without oxygen. And that's why in this tube you see cells on the top of the nutrient broth and on the bottom of the tube, because, as I said, facultative anaerobes can switch their metabolism from aerobic to anaerobic. The next group is obligate anaerobes. Obligate anaerobes do not require oxygen for growth. Even more, they can be killed by oxygen. And that's why you see all those bacterial cells on the bottom of the tube. Air-tolerant anaerobes. Air-tolerant anaerobes do not require oxygen for growth, and actually we call them air-tolerant because they don't care about oxygen. Means they cannot be killed by it, but they do not use it for metabolism. And the last group is microairfills. Microairfills require small amounts of oxygen. Not too much because they will be killed, but enough, just enough for growth. Sometimes we say those bacteria grow at the reduced oxygen. So once again, 5 groups. Obligate aerobes require oxygen for growth. Facultative anaerobes can switch their metabolism from aerobic to anaerobic. Obligate anaerobes do not require oxygen for growth. Even more, they will be killed by oxygen. Air-tolerant anaerobes do not use oxygen for growth, and they don't care about oxygen and cannot be killed by oxygen. And the last group is microaerophils, require small amounts of oxygen.